Hi, class. Today we're going to be talking about our first new pathway for the course. This is the pentose phosphate pathway. Now, the pentose phosphate pathway is a really essential metabolic pathway that provides a number of core metabolic functions, and is, as we're going to see here, is really essential for life and has direct connections to human health, and we'll be working with it quite a bit here over the next couple of weeks. So as a brief overview, the pentose phosphate pathway runs in parallel with glycolysis, and it has two primary purposes. It generates ribose for biomass, and the backbone for DNA, RNA, and it generates a reducing equivalence here in the form of NADPH. So this is the primary reducing power used both for biosynthetic reactions in the cytosol, but also as an antioxidant. So the pentose phosphate pathway has two separate stages. This first stage is this oxidative stage that we'll learn about, and the second stage is a non-oxidative stage that allows interconversion of molecules. Now, the overall equation here for the PPP is shown here, and one of the things that's unusual about it with respect to glycolysis, TCA cycle, and the other pathways that you've already learned about is that it doesn't have one-to-one stoichiometry. The pathway starts with 3 glucose-6-phosphates and 6 NADPs and 3 waters to ultimately make 6 fructose-6-phosphates, a glyceraldehyde 3-phosphate, 6 NADPHs, 6 protons, and 3 CO2s. So we're going to go through the chemistry and see how that all works. So as an overview here are the two stages of the pathway we discussed: the oxidative portion of the PPP and the non-oxidative portion. Now, an important piece of information to keep in mind is that the PPP has 3 major touch points with glycolysis. One is here at this glucose-6-phosphate stage, where carbon can enter the oxidative portion of the pathway irreversibly. It also has these 2 touch points here at fructose-6-phosphate and glyceraldehyde 3-phosphate. Now, these molecules freely flow in and out of the non-oxidative portion of the PPP, and these reactions happen at equilibrium. So I really like this particular representation of the PPP because it shows all of the fates of all the different carbons through the oxidative portion of the pathway right up here, and through the non-oxidative transketolase and transaldolase reactions. So we're going to be going through this step at a time. And the very first step that we're going to start with here is at the rate limiting step here of glucose-6-phosphate dehydrogenase. This is the rate limiting step for the oxidative portion of the PPP, and is the portion where carbon is committed in, and we get our first oxidation event generating this reduced NADPH. So here I've shown the 3 steps that are ultimately generating ribulose 5-phosphate. The first step, as I mentioned, is this glucose-6-phosphate dehydrogenase reaction, making an NADPH. That goes through this intermediate, the 6-phosphogluconolactone, that is then opened up with lactonase. This is actually a bifunctional enzyme that does both of these steps and ultimately makes 6-phosphogluconate. Now, the thing that you're going to see is really different about this from sugars that you've seen before is that this has a carboxylic acid at the end rather than an aldehyde. And what's the logic of this pathway? Putting this carboxylic acid at the end sets up a condition where this guy is just about ready to leave as a carbon dioxide. And so if you can imagine in a step here with a base abstracting a proton here, if those electrons resonate back onto this carbon, then this guy can leave as a carbon dioxide. So that's ultimately what's providing the free energy driving power that leads to this the production of this NADPH. Okay. So in these 3 steps that we're showing here, we've transitioned from glucose-6-phosphate to ribulose 5-phosphate via these 3 steps here. So the end product here, of course, so this is ribulose 5-phosphate. This is the end of the oxidative portion of the pathway. And at this point, you will have both a precursor that can go into ribose, but you'll also have all your reducing power for biosynthesis and antioxidant power. Now, there are two fates of ribulose 5-phosphate. Inside the cell, it can either be converted into ribose 5-phosphate, which of course is the sugar that's in the backbone of DNA and RNA, or it can become it can be converted by phosphopentose epimerase into xylulose 5-phosphate. So we have two enzymes here to keep track of: phosphopentose isomerase and phosphopentose epimerase. Okay, whether it's making an isomerization or an epimerization reaction. Okay, and as it turns out, the PPP is going to need both of these molecules to fully transition through the non-oxidative portions of the pathway. Meanwhile, this molecule here is getting siphoned off and going into biosynthesis. Okay, and as a brief reminder, if you don't know, an epimer is an isomer that differs only by one chiral center. So we have a change in structure here that affects chirality. Okay. Now let's take a closer look here at using that same pathway diagram I showed you earlier here at the flow of carbon through this non-oxidative portion of the pathway. So here we have our ribose 5-phosphate, which we just showed, and xylulose 5-phosphate here. And then these guys can be converted through a series of steps here that ultimately makes glyceraldehyde 3-phosphate and fructose 6-phosphate, which can enter back into glycolysis. Okay, so I'm going to go through each of these steps individually so we talk about them more. But just bear in mind that these reactions are progressing this direction, and later when we work on our problems involving carbon tracing through this pathway, come back and notice how these guys are bolded. You can see how they become these carbons here, and these guys are in the sort of darker line, and they end up here. Notice that? Now we're going to go through the other steps. So using this combination of sort of this different font and different line widths here, you can track how these two carbons end up in this position, these carbons end up in this position, and these three are originating from there, and so on and so forth. So you can track the carbon through in each position of each molecule. Okay, so now let's look a little bit here at what happens to convert these molecules from one position to another. So there are actually three reactions that happen in that occur in this non-oxidative branch. There's a transketolase reaction, there's a transaldolase reaction, and there's another transketolase reaction that's actually catalyzed by the same enzyme as this first step. Okay, and all these things are occurring in equilibrium. So even though we're sort of drawing and thinking of this pathway as flowing in this direction, it actually moves easily back and forth. So what does this all mean? So a transenolase reaction takes two 5-carbon compounds, and then, this transketolase reaction takes these two 5-carbon compounds, ribose 5-phosphate and xylulose 5-phosphate. It makes a 3-carbon compound plus a 7-carbon compound. Now notice 7 plus 3 is 10. 5 plus 5 is 10. This is how you can keep track of, whether you got all the carbons accounted for. Okay, the 3-carbon compound in this case is glyceraldehyde 3-phosphate, and we have cetaheptylose 7-phosphate that's also being made. Okay, now in the transaldolase reaction, we have this 7-carbon compound and 3-carbon compound becoming a 6-carbon compound, fructose 6-phosphate, and a 4-carbon compound with this erythrose 4-phosphate. And then in the last step here, which is also catalyzed by transketolase, we have a 4-carbon compound here, of course, erythrose phosphate, and a 5-carbon compound, fructose 6-phosphate, ultimately becoming xylulose 5-phosphate and glyceraldehyde 3-phosphate. Okay, this molecule here and this molecule here can ultimately be pulled out into glycolysis. All right, so overall, this pathway converts 3 pentose molecules in C5 form to 2 hexoses of C6 and 1 triose. So let's do it step by step. The transketolase reaction will take these 4 carbons here and split them such that the carbon, this carbon ultimately ends up on the top position of acetylheptose 7-phosphate. So added up here onto this end position of ribose 5-phosphate, and then leaves us 3 carbons behind. The second reaction here will take glyceraldehyde 3-phosphate and with this acetylheptose 7-phosphate, and then we get this intermediate here where these 3 carbon positions end up here, and this guy gets split such that we have the these guys remaining up on the top. In the erythrose 4-phosphate and xylose 5-phosphate reactions, we have these 2 guys combining here that ultimately make this fructose and glyceraldehyde 3-phosphate. And here is the table that goes through each of these reactions here, tells you the enzyme and the reaction chemistry for each. So now that we've talked a little bit about how the reactions progress from one step to another, we can talk about how carbon actually flows through the system. So unlike glycolysis, there are multiple modes of how carbon can enter and flow through this, this pathway. Now, there is this condition here. If the cell needs to make ribose 5-phosphate, but it doesn't need to have NADPH, then carbon can flow in from glucose here, flow through this non-oxidative portion of the pentose phosphate pathway, and be drawn out as ribose 5-phosphate. Okay, this is a really important source of ribogenesis or ribose biosynthesis, and decouples this biosynthetic reaction from these oxidative reactions. Here's an alternative. If NADH, NADPH, and ribose are both needed and are balanced, what can happen is that the oxidative portion of the pathway can be active, making 6-phosphogluconate and ultimately ribose 5-phosphate, and this can be drawn out. Another mode this pathway can operate in is a condition where there's a need for NADPH, and that need outweighs the need for the demand for ribose 5-phosphate. In that particular condition, glucose 6-phosphate flows in, it flows through ribose 5-phosphate, and ultimately the carbon is returned to glycolysis and then can flow down the glycolytic pathway. And here's the final condition here. If NADPH and ATP needs are balanced, then carbon will enter both glycolysis and the PPP, and so we'll get ATP biosynthesis from flux that flows through this direction, and we'll get our NADPH production from carbon that flows in this direction. Ultimately, both are flowing to pyruvate. Okay, so we've talked about different modes of how this pathway is used. Now, let's talk a little bit about what's the fate of these cofactors that get made in the pathway. So one thing that's important is being around oxygen is kind of bad for cells in general. It makes these oxidizing molecules like hydrogen peroxide and other reactive oxygen species that damage our membranes. And the primary molecule your body uses to fight this oxidation is called glutathione, or GSH. It has 2 forms. It has a reduced form, which is GSH, and an oxidized form, which is GSSG. And what that is this molecule here. And you see how it has this thiol disulfide bond right here between these 2 thiols. And these can either be broken apart via reducing them, or they can be formed through oxidation, okay? And it's the forward and back motion here between this oxidized and reduced form of glutathione that ultimately allows us to keep a redox balance inside our cells, okay? And the way these reactions occur is that, there are free radicals that are occurring from, from oxygen being around, and there's a enzyme called glutathione peroxidase that ultimately converts this hydrogen peroxide into water, and in the process takes reduced glutathione and makes oxidized glutathione. So in a separate and independent step, this oxidized glutathione is converted back into the reduced form using this glutathione reductase, which is a riboflavin-dependent enzyme, and that enzyme will ultimately take NADPH from the pentose phosphate pathway and convert it into NADP. So this pathway is really important, really, really important. It provides this biosynthetic driving power for redox driving power that's used to make biomass. And that is a really, this is a really important activity for cancer cells and other kinds of cells too. And so rapidly dividing cells frequently have a strong demand for this pathway, both to produce the biomass equivalents needed for new DNA and RNA, but also to fight oxidative damage that comes along with that metabolic activity. As it turns out, is also really important for infectious diseases, and we're going to be learning about malaria here in our tutorial sections. But malaria, as it turns out, the malaria parasite is quite sensitive to oxidative damage, and the function of the PPP can have a really profound effect on whether the malaria parasites are able to establish infections or not. So as an overview for much of the material we're going to be covering in our tutorials, malaria is this eukaryotic pathogen. It's from the genus Plasmodium. These are apicomplexan parasites, or funky parasites that are ultimately derived from these plant-like organisms. They have remnant chloroplasts in them, and they have these triple membrane bound organelles that come from the endosymbiosis of an endosymbiont. So they have this crazy evolutionary history, and these plant-based organelles in them. And they're one of our oldest diseases. Like there have been examples of sporozoites from malaria that have been found in the salivary glands of mosquitoes from the dinosaur ages. So these are very old organisms. They've co-evolved with us. They've been around ever since humans have been humans. And also, like, there are malaria parasites that affect rodents, that affect birds, that affect all kinds of lizards, all kinds of other organisms. So we're not the only organism that suffers from malaria infection. So malaria has a really complex life cycle. It's sort of beyond the scope of this course, other than to say that there are these blood phases here that undergo this 48-hour cycle of replication. And those are what are ultimately picked up by, after these things mature into gametocytes, are actually picked up by a circulating mosquito, and the parasites grow in the midgut of the mosquito, ultimately go to the salivary glands, get injected as sporozoites into people's blood, and they make the path down to the liver, where they grow and eventually burst out as new infectious sporozoites. So this is sort of a very brief life cycle that reminds you that there are mosquito phases, liver phases, and erythrocytic phases here that the parasite goes through. Now, parasite metabolism is wild, as they've undergone millions of years of evolution, and they live their entire life with a condition where they have free access to nutrients. And as a result, they've undergone an awful lot of metabolic streamlining. They've lost many of their biosynthetic pathways, so they can pick up the amino acids and nucleotides from their host cell. However, they are very dependent on a functional pentose phosphate pathway in glycolysis. So glycolysis provides all the energy for the cell. They don't have a functional oxidative phosphorylation mechanism. And they're heavily dependent on the pentose phosphate pathway, because if they lose this, they will die very quickly. They're very sensitive to oxidative damage. So this was discovered in the 1940s during these prisoner trials that in World War II, they were looking for, the Americans were experimenting with prisoners to find new drugs to fight and combat malaria. And they were experimenting with primaquine and other drugs that have these oxidative loads on people. As it turns out, some of these people who were of African descent ended up having hemolytic anemia and other really severe reactions to primaquine, other of the drugs that they were working with. As it turns out, these people had a deficiency for glucose 6-phosphate dehydrogenase. This wasn't known up until those studies that this thing existed, because without some sort of oxidative load, which can also come from things like fava beans and other kinds of oxidative stresses, this mutation doesn't have much of an outward effect on people. And so it was originally discovered, this mutation was discovered as an adverse reaction to malaria drugs. But interestingly, as it turns out, this mutation was actually evolved as a anti-malaria mechanism. And the reason for that is by having a deficiency in glucose 6-phosphate dehydrogenase, the cell actually has a lower capacity to produce NADPH. And that results in the cell oxidizing. And as it turns out, the parasites are more sensitive to that oxidative damage than cells are, and they die. So this property has been used by people making new anti-malarial drugs in the past. And this is a drug here that was used historically as an anti-malarial drug, methylene blue. So methylene blue, when it's reacted with when it reacts with oxygen, will have this blue color, and when it gets reduced back, it's colorless. Okay, so it's got this addition and loss of a hydride that changes it from this colorless to blue form. Anyway, this thing acts as a direct electron acceptor from NADPH. So NADPH can dump its electrons directly onto this blue version of the molecule and make the colorless version of it. And so this, as it turns out, is a really potent stimulator of the pentose phosphate pathway. And so as a result, it can suck up all the available NADPH, and the cell responds by shuttling a bunch more carbon through the pentose phosphate pathway. So this is a classic pentose phosphate pathway stimulator. So this has been used historically as an anti-malarial drug. Okay, to wrap up, we talked some about the pentose phosphate pathway. We discussed that it has oxidative and non-oxidative reactions, and we went through that chemistry. And we discussed how the pentose phosphate pathway is essential for many biological model systems, including malaria and cancer cells. We're going to be learning more about some of that as we move on. And yeah, welcome to our first new pathway of the course.